Right, this video is for Biology 234. We're going to be discussing differential media. So we'll go over the introduction. As you've already found, microscopy provides a limited picture of bacterial characteristics. Staining in microscopy tells us a little about the metabolism and genetics of a bacteria. In order to compare and differentiate among the enzymatic processes of bacteria, different media such as selective and differential media are used to detect metabolic and physiological differences. Selective media contain ingredients that suppress the growth of some types of organisms and allow a different type of organism to grow. So, for example, ingredients of McConkey's agar suppress the growth of gram-positive cells while selecting for growth of gram-negative bacteria. Selective media are interpreted with regard to the presence or absence of growth, where differential media use an altogether different strategy. Differential media contain reagents that will change color or react due to the growth or metabolism of bacteria. For example, the reagents in McConkey's agar will turn bacterial colonies pink if they're able to ferment lactose. Differential ingredients do not suppress the growth, they react in response to the growth. Some media, such as McConkey's agar, are both selective and differential. When using media that are selective and differential, it's important to remember that the selective assay takes precedence over the differential assay. For example, if an organism does not grow on McConkey's agar, no conclusions can be drawn regarding lactose fermentation. So, mannitol salt agar. MSA is a selective and differential media that contains the sugar mannitol, phenol red, which is a pH indicator, and 7.5% sodium chloride. MSA selects for halotolerant organisms, which are bacteria that can survive in a relatively high salt environment. MSA is differential for the ability of bacteria to ferment mannitol. If a halotolerant organism is able to ferment mannitol, the acids produced will cause the phenol red in the medium to turn yellow. Bacterial growth with no color change indicates halotolerance, but no ability to ferment mannitol. If an organism does not grow on MSA, no conclusions regarding fermentation can be drawn. MSA is used in the clinical environment to differentiate between Staphylococcus aureus and other species of Staphylococcus. Phenylethyl alcohol agar, so PEA, is selective for gram-positive bacteria. The growth of gram-negative bacteria is suppressed by the interaction of alcohol with the outer membrane. Gram-negative bacteria may not be completely inhibited, but the growth will be greatly diminished when compared to growth on TSA. Gram-positive bacteria are typically unaffected by the phenylethyl alcohol and will grow nearly as well as on TSA. McConkey's agar. MAC is a selective and differential media that contains the sugar lactose, phenol red, which is a pH indicator, bile salts, and crystal violet. MAC is selective for gram-negative bacteria. The bile salts and crystal violet inhibit the growth of gram-positive bacteria when compared to a TSA control. If a gram-negative organism is able to ferment lactose, the colonies will turn pink and may have a haze around them. Gram-negative organisms unable to ferment lactose will be colorless. The ability of an organism to ferment lactose can only be determined if there is growth. If there is no growth, no information regarding lactose fermentation can be gathered. Alright, so now we're ready to start the procedure for selective and differential media. You will inoculate plates of MSA, PEA, and MAC with your unknown, a positive control, and a negative control. So I'll gather the materials that we need. So we'll need, so these are our controls. And then this is our unknown. And then I'll grab 3 of each, the MSA, PEA, and MAC plates. So this is the PEA, this is the MAC, and then this is the MSA. So now we will label our plates before we begin. So normally it tells you to divide one plate with your unknown and then the 2 controls, but we found that often it'll grow into the other quadrant and then it's really hard to determine your results. So I'm just going to do one streak on 3 different plates so that there's no question about our results. So it's also good to just let these air dry because otherwise it's kind of hard to write on them. So I'm just gonna use a paper towel to remove any moisture. All right, so I'll start with staph epidermidis. And the date. And then your initials. But I just realized, so this is the first time we're using a different media than TSA. So you also want to write what media you're using. So this one's PEA, this one's MAC, and this one is MSA. So we'll start with the staph epidermis, the date, my initials, and then this one is MSA. So this will be all the staph epidermidis, and then I'll move on to the E. coli. And then this one will be for our unknown number 9. All right, so all of our plates are labeled and now we're ready to transfer the cultures. So this will be very similar to the 4 quadrants streak, except for we're not doing the quadrants, we're just going to do 1 streak. So we'll want to sterilize our loop. So 1, 2, 3, 4, 5. And we'll start with the staph epidermis. And then you just streak it. And then we'll sterilize their loop again. Then we'll grab some culture and then streak it onto our plate. And then we will sterilize our loop again. grab culture, and then streak it onto our plate. Then we'll sterilize our loop again. And then let that cool. We'll put these off to the side. We'll put this off to the side. So now we will do E. coli. And then I'm going to put unknown 9 by these. So we'll be ready. So now I'll sterilize my loop again. So 1, 2, 3, 4, 5. And then I'll grab some culture. This one. And then streak it onto plate. Then we'll sterilize the loop again. So 1, 2, 3, 4, 5. And then we'll grab some culture, streak it onto our plate, and then sterilize our loop again. So 1, 2, 3, 4, 5. And then I touch it to a blank spot of the auger. Grab some culture, and then streak it onto our plate, and then sterilize your loop again. So 1, 2, 3, 4, 5. And then just let that cool. And now we'll move on to unknown number 9. So we'll sterilize our loop again. So 1, 2, 3, 4, 5. Then we'll grab some culture and stick it onto our plate. And then sterilize our loop again. And then we'll grab some culture, streak it onto our plate, and then sterilize our loop again. So 1, 2, 3, 4, 5. And then we'll grab some culture, streak it onto our PEA plate, and then sterilize our loop again. So 1, 2, 3, 4, 5.